Segregation and independent assortment are key processes that occur during meiosis. Segregation refers to the separation of alleles for each gene, while independent assortment describes how alleles for one gene segregate independently of alleles for other genes. When genes are located on separate chromosomes, they are considered unlinked and undergo independent segregation during meiosis. However, genes located on the same chromosome are linked and do not segregate independently, except in cases where they are far apart on the chromosome. In such instances, crossing over between genes can occur more frequently, giving the appearance of independent segregation. Next, we are going to explore Punnett squares, a useful tool for understanding how traits are inherited. We'll cover both monohybrid and dihybrid crosses. First, let's dive into monohybrid crosses. These involve the inheritance of a single trait governed by one gene with two alleles. To create a Punnett square, we'll consider a hypothetical cross between two individuals, each carrying two different alleles. Let's take an example of a cross between a purple-flowered plant, two capital P's, and a white-flowered plant, two lowercase p's. We represent dominant alleles with uppercase letters and recessive alleles with lowercase letters. Now, we'll construct a Punnett square by placing the alleles from one parent along the top and the alleles from the other parent along the side. Each box in the Punnett square represents a possible combination of alleles that their offspring can inherit. Now, let's fill in the boxes with the corresponding allele combinations. To determine the probability of a particular phenotype, we analyze the genotypes. In this monohybrid cross, since all the offspring have the same genotype, capital P lowercase p, they will exhibit the same phenotype, which is purple flowers. Now, let's move on to dihybrid crosses, which involves the inheritance of two different traits governed by two genes. We'll extend our Punnett square to accommodate the additional gene. Imagine we are crossing two pea plants, one with round yellow seeds, two capital R's and two capital Y's, and the other with wrinkled green seeds, two lowercase r's and two lowercase y's. Here, we'll use the uppercase letters for dominant alleles and lowercase letters for recessive alleles. To construct a dihybrid Punnett square, we'll place the alleles of one seed along the top and the alleles of the other seed along the side. In this case, only one type of gamete can be made for each seed, capital R, capital Y, and lowercase r, lowercase y. However, depends on the genotype of parent generation shown in the question you encounter, this might vary. Now, let's fill the boxes with the corresponding allele combinations. As you can see, the Punnett square shows all the possible combinations of allele that the offspring can inherit for both traits. Now, let's determine the phenotypes based on the genotypes. In this dihybrid cross, when we analyze the genotypes, we find that all offsprings have a combination of round seeds, capital R, lowercase r, and yellow seeds, capital Y, lowercase y. Therefore, we'll observe a single phenotype in the offspring, which is round yellow seeds. Punnett squares provide a visual representation of the possible combinations of alleles and help us predict the inheritance patterns of traits in offspring. They are a valuable tool in understanding genetics and can be used for both monohybrid and dihybrid crosses. Remember, Punnett squares are based on the principles of segregation and independent assortment, which we discussed earlier. By using Punnett squares, we can make predictions about the genetic outcomes of crosses and gain insights to the inheritance of traits. Next, we'll explore the groundbreaking work of Gregor Mendel, known as the father of modern genetics. We'll discuss Mendel's rules and also delve into exceptions that challenged his original findings. Mendel formulated 3 fundamental rules based on his experiments with pea plants. These rules laid the foundation for understanding inheritance patterns. Let's explore each rule. Mendel's first law, the law of segregation, states that during the formation of gametes, which are reproductive cells, the two alleles of a gene separate from each other so that each gamete carries only one allele for each gene. The second rule, the law of independent assortment, states that the alleles of different genes segregate independently of one another during gamete formation. In other words, the inheritance of one trait does not influence the inheritance of another trait. Mendel's third rule, the law of dominance, states that in a cross between two individuals with different alleles for a particular gene, only one allele, the dominant allele, will be expressed in the offspring, while the other recessive alleles remains hidden. Although Mendel's rules explain many inheritance patterns, there are exceptions that challenged his original findings. Let's discuss two notable exceptions, sex linkage and linked genes. Sex linkage refers to the inheritance of genes on the sex chromosomes, X and Y. In humans, for example, the gene for color blindness is located on the X chromosome. Since males have only one X chromosome, they have a higher chance of expressing X-linked traits. Females, on the other hand, have two X chromosomes, providing a greater chance to carry a recessive allele without expressing the trait. Usually, the sex-linked disease you'll encounter in IV are related with the X chromosome, since Y chromosome is much shorter than X chromosome, containing less genes. Linked genes are genes located close together on the same chromosome. Unlike the law of independent assortment, these genes do not segregate independently. During meiosis, linked genes tend to be inherited together more frequently than not. However, the exception lies in crossing over, a process where genetic material is exchanged between the homologous chromosomes during meiosis. Crossing over can separate linked genes, leading to new combinations. Mendel's rules, segregation and independent assortment, provide a solid framework for understanding inheritance patterns. However, exceptions like sex linkage and linked genes introduce complexities that Mendel was unaware of during his time. Let's move on to the genetic variations. Genetic variations are differences that exist among individuals within a species. Today, we will explore two types of variations, continuous variation and recombinance. Continuous variation refers to a type of variation in which a trait shows a wide range of possible values across a population. These traits are influenced by multiple genes and often exhibit a bell-shaped distribution. Let's consider an example of human height. Height is a trait that shows continuous variation. In a population, you'll find individuals of various heights, ranging from very short to very tall. The height of an individual is influenced by multiple genes and environmental factors, leading to a continuous distribution of values. Continuous variation is often represented by a bell-shaped curve called a normal distribution or Gaussian distribution. The majority of individuals tend to cluster around the average height, while fewer individuals are found at the extremes of short or tall heights. It's important to note that continuous variation is not limited to height. Traits like weight, skin color, and even intelligence can also exhibit continuous variation in a population. Recombinants are a type of variation that occurs due to the recombination of genetic material. Recombination is the exchange of genetic material between homologous chromosomes during the process of meiosis. During meiosis, when cells divide to produce gametes, reproductive cells, genetic material can be exchanged between chromosomes through a process called crossing over. This exchange of genetic material leads to the creation of new combinations of alleles, giving rise to genetic variation. The offspring that inherit these new combinations of alleles, different from those of their parents, are called recombinants. Recombinants carry unique combinations of genetic traits that may not be present in either of their parents. Recombination plays a crucial role in generating genetic diversity within a population. It contributes to the shuffling and mixing of genetic information, leading to new combinations of traits and increasing the potential for adaptation and evolution. Recombinants are particularly significant in understanding genetic inheritance and mapping genes on chromosomes. By analyzing the patterns of recombinants, scientists can determine the relative positions of genes on a chromosome, helping unravel the genetic basis of various traits. In summary, genetic variations come in various forms. Continuous variation involves traits that show a wide range of possible values across a population, often represented by a normal distribution curve. Recombinants, on the other hand, result from the exchange of genetic material during meiosis, leading to new combinations of alleles and increased genetic diversity. Understanding these different types of genetic variations help us appreciate the complexity and diversity of life. It also enables us to study inheritance patterns, evolution, and the factors that contribute to the unique characteristics of individuals within a population. Lastly, we'll explore the chi-square test, a statistical tool used to analyze categorical data and determine if there is a significant association between variables. The chi-square test is a hypothesis test that compares observed data with expected data to determine if there is a statistically significant difference. It helps us understand whether the observed frequencies in different categories deviate significantly from what we would expect under a null hypothesis of independence. The test is used when we have categorical data, such as counts or proportions, and we want to examine if there is a relationship or association between two or more variables. Before performing the chi-square test, we formulate our null hypothesis H0 and alternative hypothesis H1. The null hypothesis assumes that there is no association between the variables, while the alternative hypothesis suggests that there is a significant association. To conduct the chi-square test, we start with a set of observed data, which represents the actual frequencies or counts in each category. We also calculate the expected data, which represents the frequencies we would expect under the assumption of independence. The expected data is calculated based on the null hypothesis and can be determined using different methods, such as assuming equal proportions or using specific theoretical distributions. Once we have the observed and expected data, we calculate the chi-squared statistic. This statistic measures the difference between the observed and expected frequencies, taking into account the size of the sample and the number of categories. The chi-square statistic is calculated by summing the square differences between the observed and expected frequencies divided by the expected frequencies. After calculating the chi-squared statistic, we compare it to a critical value from the chi-square distribution. The critical value is determined based on the desired level of significance, often denoted as alpha. By comparing the chi-square statistic to the critical value, we can determine if the observed data deviates significantly from what we would expect under the null hypothesis. This is done by calculating the p-value, which represents the probability of obtaining a test statistic as extreme as the observed one, assuming the null hypothesis is true. The chi-square test is a valuable statistical tool for analyzing categorical data and investigating associations between variables. It helps us determine if there is a significant relationship beyond what could be expected by chance. By comparing observed and expected data, calculating the chi-square statistic, and examining the p-value, we can make informed conclusions about the presence or absence of an association. Here's a practice question. Pause the video and try to answer this. Here's the answer. That concludes our lesson on the inheritance. I hope you found it informative and helpful.